Welcome dear viewers. In this video, I would like to explain about the zoonotic disease caused by rabies virus. I am Dr. M. Manikandan, Assistant Professor from the Department of Microbiology, Sacred Heart College, Tirupattur. Introduction. The rabies virus belongs to the family Rhabdoviridae and the genus Lysaviruses. In Greek, the rhabdos meaning rod-shaped. The rabies virus includes pathogen for a variety of mammals, fishes, birds, and plants. The family contains vesiculoviruses, that is, vesicular stomatitis viruses (VSV), and lysaviruses, that is, rabies and rabies-like viruses. Rabies virus is one of the most significant pathogen of rhabdoviruses. Louis Pasteur developed the killed rabies vaccine in 1885 from a bite of a mad dog, which always lead to the characteristic symptom of hydrophobia and certainly death. Transmission. The mode of transmission of rabies virus has demonstrated a remarkable ability to adapt to new host, which is essential for its survival and spread. The rabies virus may get transmitted from human to animal, that includes both domestic and wild animals. The domestic animals, pets and farm animals, are cats, cows, dogs, ferrets, goats, and horses. The wild animals includes bats, beavers, coyotes, foxes, monkeys, raccoons, skunks, and woodchucks. Structure. Rhabdoviruses are simple viruses encoding only 5 proteins and appearing as bullet-shaped, enveloped, helical-shaped virions with a diameter of 50 to 95 nanometer and length of 130 to 380 nanometer. Spikes composed of a trimer of the glycoprotein G, which covers the surface of the virus, which also acts as the viral attachment protein, and the G protein generates neutralizing antibodies. Within the envelope, the helical nucleocapsid is coiled symmetrically into a cylindrical structure, giving it the appearance of striations. The nucleocapsid is composed of one molecule of single-stranded negative sense RNA of approximately 12,000 bases with the N protein, L protein, and NS protein. The M protein lies between the envelope and the nucleocapsid. The N protein is the major structural protein of the virus. It protects the RNA from ribonuclease digestion and maintains the RNA in a configuration acceptable for transcription. The L protein and NS proteins constitute the RNA-dependent RNA polymerase enzyme. Replication of the rabies virus. The viral G glycoprotein attaches to the host cell surface receptor and is entered into the host cell by endocytosis. The rabies virus binds to either the nicotinic acetylcholine receptor AChR or neural cell adhesion molecule NCAM or by other molecules. The viral envelope then fuses with the membrane of the host cell endosome on acidification of the vesicle. This uncoats the nucleocapsid, releasing it into the cytoplasm where the replication takes place. Endosomal vesicles may deliver whole rabies virions along the axon to neuronal cell bodies where its replication takes place. The RNA-dependent RNA polymerase associated with the nucleocapsid transcribes the viral genomic RNA, producing 5 individual mRNAs. For rabies virus, this occurs in the Negri bodies. These mRNAs are then translated into 5 important viral proteins. The viral genomic RNA is also transcribed into a full-length positive sense RNA template that is used to generate new genomes. Assembly and release of the rabies virus. The G glycoprotein is synthesized by membrane-bound ribosomes, processed by the Golgi apparatus, and delivered to the cell surface in membrane vesicles. The assembly of these virions occurs in two different phases. The first one is the assembly of the nucleic capsid in the cytoplasm and envelopment and release at the cell plasma membrane. The virus then buds through the plasma membrane and is released when the entire nucleic capsid is enveloped. Cell death and lysis occurs after infection with most rhabdoviruses, with the important exception of rabies virus, which produces little discernible cell damage. This is the diagrammatic representation of the pathogenesis of rabies virus, starting from the virus inoculation through the bite of a rabid dog, and then virus replication occurs at various sites, and then it moves towards the spinal cord, and then disseminate. The viral particle will disseminate after the replication in the central nervous system to the various sites, including eye, salivary glands, skin, and other organs. The pathogenesis route of a rabies virus. Rabies infection usually results from the bite of a rabid animal. Rabies infection of the animal causes secretion of the virus in the animal's saliva and promotes aggressive behavior, that is mad dog, which in turn promotes transmission of the virus. The virus can also be transmitted through the inhalation of aerosolized virus, for example, that is the viral transmission from bat caves, and then in transplanted infected tissues, example cornea, and by inoculation through intact mucosal membranes. The virus replicates quietly at the site of infection or the site of entry for days to months before progressing to the central nervous system. Rabies virus travels by the retrograde axoplasmic transport to the dorsal root ganglia and to the spinal cord. Once the virus gains access to the spinal cord, the brain becomes rapidly infected. The affected areas are the hippocampus, brainstem, ganglionic cells of the pantine nuclei, and Purkinje cells of the cerebellum. The virus then disseminates from the central nervous system via afferent neurons to highly innervated sites such as the skin of the head and neck, salivary glands, retina, cornea, nasal mucosa, adrenal medulla, renal parenchyma, and pancreatic acinar cells. After the virus invades the brain and spinal cord, encephalitis develops and neurons degenerate. Despite the extensive CNS involvement and impairment of the CNS function, little histopathologic changes can be observed in the affected tissue, other than the presence of Negri bodies. Clinical syndromes. Rabies infection is virtually always fatal unless treated by vaccination. The incubation period in human varies from 1 to 3 months, but it may be as short as 1 week to as long as many years. It is usually shorter in children than in adults. The clinical spectrum of rabies can be divided into several phases. The first one is the prodromal phase, which lasts for 2 to 10 days. The symptoms are fever, malaise, headache, pain or paresthesia, that is itching at the site of bite, gastrointestinal symptoms including fatigue and anorexia. After the prodromal phase, the neurological symptoms specific to rabies appear. It lasts for 2 to 7 days. The infected individual will have nervous system disorders like nervousness, apprehension, hallucination, lacrimation, pupillary dilation, and increased salivation with hydrophobia and painful throat, throat muscles while swabbing. The most important symptom of rabies, that is hydrophobia, fear of water, which occurs in 20 to 50 percentage of the infected patients. It is triggered by the pain associated with the patient's attempt to swallow water. 15 to 60 percentage of patients exhibit paralysis as the only manifestation of rabies. The paralysis may lead to respiratory failure. After the neurological phases, coma, the third phase, coma and death may occur. The patient becomes comatose after the neurological phase, which lasts for 2 to 10 days. This phase almost usually leads to the death resulting from neurologic and pulmonary complications. Laboratory diagnosis. Laboratory tests are usually performed to confirm the diagnosis and to determine whether a suspected individual or animal is rabid. The specimen to be collected at various stages are acute mortem, and the samples are hair follicles and saliva. And after the post-mortem, the samples to be collected are salivary gland, brainstem, hippocampus, and cerebellum. Antigen detection using direct immunofluorescence test or genome detection using reverse transcriptase polymerase chain reaction, that is RT-PCR, are relatively quick and sensitive assays that are preferred methods for diagnosing rabies. The samples to be collected for this test are saliva, which is easy to be utilized. Rabies can also be grown in cell culture or in intracerebrally inoculated infant mice, but it requires special laboratory isolation procedures and is not routinely performed. Inoculated cell cultures or brain tissues are subsequently examined with direct immunofluorescence test. The rabies antibody titers in serum and cerebrospinal fluid are usually measured by enzyme-linked immunosorbent assay, or a rapid fluorescent focus inhibition test may also be used. Antibody is usually not detectable until the late phases of the disease. The hallmark diagnostic finding has been the detection of the intracytoplasmic inclusions consisting of aggregates of viral nucleocapsid, that is Negri bodies, in affected neurons. Negri bodies. These are the pathological diagnostic materials first observed by Sir Adolphe Negri in 1903 in infected rabbits and dogs' brain tissues. The Negri bodies are eosinophilic inclusion bodies found in the cytoplasm of certain nerve cells infected with the rabies virus. The Negri bodies contains viral ribonucleoprotein with peripheral fully formed virions budding into the cytoplasm. They are typically 2 to 10 micrometer in diameter and observed in the pyramidal cells of the hippocampus and Purkinje cells of the cerebellar cortex in the postmortem brain samples from rabies victims. Treatment and prevention of rabies virus infections. The first protective measure is local treatment of the wound. The wound should be washed immediately with soap and water or any other substance that inactivates the virus. The World Health Organization Expert Committee on Rabies also recommends the installation of anti-rabies serum around the wound. Subsequently, immunization with vaccine in combination with administration of one dose of human rabies immunoglobulin (HRIG) or equine anti-rabies serum is recommended. Passive immunization with HRIG provides antibody until the patient produces antibody in response to the vaccine. The rabies vaccine is a killed viral vaccine prepared through the chemical inactivation of rabies infected tissue culture human diploid cells, that is HDCV, or fetal rhesus lung cells. These vaccines causes fewer negative reactions than the older vaccine, that is simple and Fermi vaccines, which were prepared in the brains of adult or suckling animals. The HDCV is administered intramuscularly on the day of exposure and then on days 3, 7, 14, and 28, or intradermally with a lower dose of vaccine to multiple sites on days 0, 3, 7, 28, and 90. Pre-exposure vaccination should be performed on animal workers, laboratory workers who handle potentially infected tissue, and people traveling to areas where rabies is endemic. HDCV administered intramuscularly or intradermally in 3 doses is recommended and provides 2 years of protection from the rabies virus. Thank you, dear viewers. Kindly subscribe to our channel and help us improvise by your comments. Once again, thank you.